Meiosis, starring Melanie McCormick
and Meredith Frost. Hi, I'm Brad Pitt.
And I'm Auntie Jolie. And we're having a baby.
What did you say?
Yay!
Woohoo! How?
It's simple. By meiosis. Duh.
What's meiosis?
Brad, you're so funny. Well, it's a type of cell division that produces a haploid gamete in a diploid organism, such as us humans.
Ooh, how does it start?
First off, the cell grows and replicates DNA in a phase called interphase.
Cool. What happens next?
Next, the cell is signaled to begin meiosis. Meiosis has two parts, meiosis 1 and meiosis 2. In meiosis 1, which is when homologous chromosomes separate, prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis take place.
Prophase 1 is the most complex phase of meiosis and typically occupies over 90% of the time required for meiotic cell division. Early in this phase, the chromatin coils up so that individual chromosomes become visible with a microscope. In a process called synapsis, homologous chromosomes, each composed of two sister chromatids, come together as pairs. The resulting structure, consisting of 4 chromatids, is called a tetrad. During synapsis, chromatids of homologous chromosomes exchange segments in a process called crossing over. Because the versions of the genes on a chromosome or one of its chromatids may be different from those on its homologue, crossing over rearranges genetic information. The genetic shuffling produced by crossing over can make an important contribution to the genetic variability resulting from sexual reproduction. As prophase 1 continues, the chromosomes condense, further as the nucleoli disappear. Now the centrosomes move away from each other and a spindle starts to form between them. The nuclear envelope breaks into fragments, and the chromosome tetrads, captured by spindle microtubules, are moved toward the center of the cell.
At metaphase 1, the chromosome tetrads are aligned on the metaphase plate, midway between the two poles of the spindle. Each chromosome is condensed and thick, with its sister chromatids still aligned at centromeres. Spindle microtubules are attached to kinetochores at centromeres. In each tetrad, the homologous chromosomes are held together at the sites of crossing over. Notice that for each tetrad, the spindle microtubules attached to one of the homologous chromosomes comes from one pole of the cell, and the microtubules attached to the other homologous chromosome come from the opposite pole. With this arrangement, the homologous chromosomes of each tetrad are poised to move toward opposite poles of the cell.
Anaphase 1 of meiosis is marked by the migration of chromosomes toward the two poles of the cell. In contrast to mitosis, however, the sister chromatids making up each doubled chromosome remain attached at their centromeres. Only the tetrads, pairings of homologous chromosomes, split up. Thus, in the drawing you see 2 still doubled chromosomes, the haploid number, moving toward each spindle pole. If this were anaphase of mitosis, you would see 4 daughter chromosomes moving toward each pole.
In telophase 1, the chromosomes arrive at the poles of the cell. When the chromosomes finish their journey, each pole of the cell has a haploid chromosome set, although each chromosome is still in duplicate form at this point. In other words, each chromosome still consists of 2 sister chromatids. Usually, cytokinesis occurs along with telophase 1, and 2 haploid daughter cells are formed. Following telophase 1 in some organisms, the chromosomes uncoil and the nuclear envelope reforms, and there is an interphase before meiosis 2 begins. In other species, daughter cells produced in the first meiotic division immediately begin preparation for the second meiotic division. No chromosome duplication occurs between telophase 1 and the onset of meiosis 2.
In organisms having an interphase after meiosis 1, the chromosomes condense again and the nuclear envelope breaks down during prophase 2. In any case, meiosis II is essentially the same as mitosis. The important difference is that meiosis II starts with a haploid cell. During prophase II, a spindle forms and moves the chromosomes toward the middle of the cell. During metaphase II, the chromosomes are aligned on the metaphase plate as they are in mitosis, with the kinetochores of sister chromatids of each chromosome pointing toward opposite poles. In anaphase II, the centromeres of sister chromatids finally separate, and the sister chromatids of each pair, now individual daughter chromosomes, move toward opposite poles of the cell. In telophase II, nuclei form at the cell poles and cytokinesis occurs at the same time. There are now 4 daughter cells, each with the haploid number of chromosomes.
So, Brad, now do you understand?
Yeah, but what does that have to do with making a baby?
Well, these diploid cells that meiosis produces come together and form a zygote, which turn into our baby.
Yeah, let's do it!